right, welcome to this lecture. We're going to be finishing up our topics 1.3 to 1.4 discussing cellular membranes. And just to review, prior to this, we've been talking about diffusion and osmosis. And so just as a reminder, diffusion is the movement of any type of molecule with a concentration gradient. So in that case, it goes from higher concentration to lower concentration. And osmosis is the movement of water from an area of lower solute concentration to higher solute concentration. So water moves to dilute, and that's a really important distinction. For diffusion, you can say moves from high concentration to low concentration. But for osmosis, you have to make the distinction that it's water moving depending on what's dissolved in it. So be sure that you are making that distinction when you discuss diffusion and osmosis. Today we're going to be talking about other ways that atoms can move through a cellular membrane. And so let's get started. Okay, the first thing we're going to talk about is simple and facilitated diffusion. This type of movement requires no energy, and simple diffusion is basically what we've already been talking about. There's no energy, and the molecules are simply moving across the plasma lipid bilayer, and they're moving from an area of higher concentration to an area of lower concentration, and they are just moving straight through those phospholipids. So no proteins, no nothing, no energy, just moving through. On the other hand, we have facilitated diffusion. Now, this type of movement still requires no energy, so it's still moving from high concentration to low concentration, but this is movement that occurs when either a molecule is too large or a molecule is charged and so will not move through the fatty acid tails of the phospholipids due to the polar-nonpolar relationship. So if a molecule is too large or if it has a charge, it's going to move by a way called facilitated diffusion, which means that it moves through a channel protein, as you can see here, that basically just remains open and allows a path through the lipid bilayer. So no energy needed. The only difference is instead of going straight through the plasma membrane, the molecule is going to be moving through a channel protein, which also, as you can see, is an integral protein because it goes all the way across the lipid bilayer. Our last two forms of movement do require energy. So these are cases where instead of moving with the concentration gradient, so from higher concentration to lower concentration, you are moving against that gradient. So in these cases, the movement doesn't happen naturally, therefore energy is needed. The first type of movement we'll talk about in that category is active transport. And in this case, like I said, molecules are moving from low to high concentration, therefore must be actively moved. And in this case, you're using an integral protein, one that goes all the way through the lipid bilayer. But instead of being a channel protein, it is a pumping protein. And so as you can see here, the molecule is going to be loaded into the pump. ATP is going to be used, and by being used, what that means is that one phosphate is removed from the ATP molecule. This causes a change in the shape of this pump protein, so now it's open to the outside of the cell, and then the molecule can be released. And so as you can see, in this case, the inside of the cell has a lower concentration and we're moving to an area of higher concentration. So energy must be expended for this to happen. Real quickly, we're going to actually watch another video that animates this process.
In this case, the cell uses transport proteins which need a boost from an energy molecule. This actually changes the shape of the proteins, causing them to pump different molecules and ions across the membrane. Now from the side with fewer solutes to the side with more.
All right, so that was active transport. The last type of transport we're going to be looking at is called cytosis. And this is an active process. It does require energy, And this is a situation where we've got a number of large molecules that need to move in a couple of different ways. Number one, they could be moving between different organelles within the cell. And remember, organelles in a eukaryotic cell are surrounded by a plasma membrane. And so moving from one to the other requires a change in that membrane. Or number two, this could be the movement of molecules or different things into or out of the cell itself. Okay, so in this situation, the molecules being moved are going to be too large for simple or facilitated diffusion or active transport. These are going to be things like proteins, fats, things that the cell has produced that need to travel elsewhere. Okay, so we've got two types of cytosis. We've got exocytosis and endocytosis. Exocytosis is a situation where items are leaving the cell. And so in this case, the items that need to leave are bundled into what's called a vesicle, which is basically just a membrane-bound sac. And that membrane-bound sac fuses with the plasma membrane, and then it opens up to the outside of the cell, releasing the contents of that vesicle. Okay, the opposite of this is endocytosis, and that is a process where items are being brought into the cell. And so in this case, the items that need to enter the cell come near the plasma membrane. The plasma membrane will then make kind of an indent. It looks almost kind of like a Pac-Man situation. And the items that need to come in here are going to then be engulfed by this plasma membrane and brought into the cell to be transported to wherever they need to go. All right, so here is an image, a light microscope image of this process actually happening. And in this case, what we're looking at is endocytosis. So here are the items that need to come into the cell. A little pocket is being formed. That pocket is closing, making a vesicle that travels into the cell. So in this case, what we have is the phospholipid molecules are able to change places horizontally, but not vertically. So remember, we have a bilayer. They can kind of move around with their neighbor, but they're never going to flip places with the phospholipid across from them. So what this process does is it allows the cell to bring contents in and have contents exit without ever making a hole or a space in the cell so that things that aren't meant to come in can come in. So I have a video that animates this that we'll watch now.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane. This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
All right, so please take a minute, pause the video if you need to, and write down any questions that you have about any of these transport systems, whether simple or facilitated diffusion, active transport, or cytosis. It's really important that you understand all of these. Over the next couple days, you guys are going to be doing a lab that looks at the principle of osmolarity. So This little prompt says tissues or organs being used in medical procedures must be bathed in a solution with the same osmolarity as the cytoplasm to prevent osmosis. So what this is saying is when you're doing an organ transplant, that organ needs to be bathed in a solution to keep the cells moist, to provide nutrients, all the things the cell needs for the period that it's not being supplied by the body. But the solution that it's in must be the same osmolarity as the organ itself. Otherwise, water will get sucked out of the organ, or water will get sucked into the organ, and the organ could swell and rupture. So what we're going to be doing is you'll be given an unknown substance. It's actually going to be a potato. And you're going to be bathing it in different samples of sucrose solution to determine what is the osmolarity of the potato. And so tomorrow you are going to be doing the exploration phase. But today, I want you to do a little research and come up with a procedure. How are you going to do this? And when you do it, how are you going to tell what the osmolarity of the potato is? All right, so take some time, get some research done today, and you'll be doing your exploration phase tomorrow. Lastly, I just want to give credit to the other videos that were inserted within my own video. So here are the citations for those. Check out these channels. They've got some awesome short animations about cells. Thanks. Have a great day.